Well, I would like to thank OIS for allowing us to present our pipeline and introduce our company, Allgenesis. So our goal and our core mission is to develop next-generation therapeutics to treat age-related macular degeneration, diabetic macular edema, and dry eye diseases. We got two molecules, AG-73305 and AG-8038. Our senior leadership team and management team, we have combined 200 years of drug development experience. And our founding partners are Dr. Chen and Dr. Cherukuri. So retinal diseases, the current standard of care is just not effective enough in modifying disease state. As noted that 2 out of 3 patients are partial non-responders to anti-VEGF monotherapies. And there's no molecule that causes regression of CNV. AG-73305 is a bifunctional Fc fusion protein, which I'll describe additionally. And the market share we're talking about for DME and neovascular AMD is expected to reach about $18 billion in 2025. So our core molecule is AG-73305. It's a bispecific design, provides added benefit over anti-VEGF monotherapies. The proximal end is a aflibercept. It acts like aflibercept and is just as potent as aflibercept, binds VEGF-AB and placental growth factor. The business end of this molecule, in addition to the signaling pathway, is the anti-integrin. And this is a pan-integrin or anti-integrin antagonist. It blocks the cells responsible for neovascularization, inflammation, fibrosis, inhibits IL-6, IL-1, and TNF-alpha. And so we're expecting to benefit both anti-VEGF responders and non-responders in DME. So the goal of this molecule is it blocks the signaling pathway through the anti-VEGF early on, and then the anti-integrins, which play a role, or integrins play a role in cell-cell-cell ECM interactions, it'll block that to give us additional durability. And by blocking the integrins, we'll modify the disease state. As you know, integrins are really important in retinal diseases. So blocking the integrins would alter the disease state itself. And in choroidal neovascularization, alpha V, beta 3, and beta 5 are important, as well as vascular permeability, retinal inflammation, retinal fibrosis, and our molecule targets all of them. So this is not strictly a signaling pathway inhibitor. It's actually inhibiting the effector pathway. So when we're inhibiting using TKIs or Ang2 or IL-6, they're still using the integrins on the downstream effector pathway. So we're going out and blocking those directly. So it's the action pathways that cause the disease state is where our molecule works, in addition to the anti-VEGF component. So we did do a phase 2A study on diabetic macular patients for AG-73305. It was just a single dose escalation study of 0.5, 1, 2, and 4 milligrams. And for DME, the inclusion criteria was 325 microns and BCVA 2040, 2400. And the primary endpoints were to assess safety and then BCVA and SD-OCT for CST. Baseline demographics, a small study, but it was unbalanced, but the mean vision ranged from mean age range from 55 to 70. And the ocular characteristics, mean BCVA ranged from 49 to about 63. And the CST was 445 to 658. So it's quite a bit of range, but we're looking at 3, 6, 8, and 8 patients for a total of 25. But we're just trying to see how our molecule is working. So looking at this, basically, we did not see any safety issues at all. There were no retinal vascular issues, vasculitis, retinitis, occlusive vasculitis, or vitritis. We did see some anterior chamber inflammation, which was well controlled with topical steroids, and it resolved within 1 to 2 to 3 weeks. As you can see, at the 0.5 milligram dose, this is a single dose, we're seeing an improvement of BCVA up to 8 weeks, and is maintained for 24 weeks in these patients. 1 milligram dose was much better with the BCVA of 9 letters, and that continued up to 24 weeks as well with the durability. And 2 and 4 were also beneficial, and we were able to maintain the BCVA for 24 weeks. CST was somewhat similar. And as you can see, 1 milligram was better than the 0.5, 2, and 4, But again, we were able to see the CST decrease with the treatment. These are just a couple examples of patients with, this is a single 1 milligram dose. And at baseline with the heat map, it's pretty thickened retina, but over 4, 8, 16, 20 weeks, there's continuous improvement in the CST with the single dose. So anti-VEGF treatments would pretty much wear off at 4 to 8 weeks. We're seeing a sequential improvement up to 24 weeks. This is another patient, somewhat similar, where you got significant thickening with the heat map, and at up to week 24, it's just continual improvement of CST and improvement of the BCVA. This is a Kaplan-Meier, and we're just looking at how often they needed to be rescued or how quickly, and we were able to maintain non-rescued patients up to 50% for up to 24 weeks. So it's the durability, we're seeing a really good durability signal. So in summary, this is our molecule is safe with no posterior segment inflammation, no dose-limiting toxicity, mild AC inflammation, which was well controlled with topical steroids. BCVA improvements were maintained up to 4 to 6 months, and anti-ingredients domain may play a role in modifying the disease state. And so we got a compelling evidence. We're going to go ahead and proceed to probably a 2B comparator trial. Our project pipelines and key milestones, we're looking at doing a phase 2B for DME, as well as neovascular AMD, which we haven't done, but we want to start that with retinal occlusion as well as PVR. And we have completed the phase 1B for our dry eye molecule, and want to partner with good partners here. We've raised so far $50 million, looking to raise another $25 million for our CMC activities and bridging study, and then obtain another $25 million to advance project to next milestones for IPO, which is planned in 2025. Again, thank you very much. Appreciate you guys and OIS for allowing us to present our data. Thanks.